Good morning everyone. So in my first video I talked about overview of fatty acid synthesis. In my second video I talked about steps of fatty acid synthesis. This is my last video on fatty acid synthesis where I'm going to complete the steps and also talk about regulation. Before I complete this pathway, let's do a quick recap of what we have learned so far. So as you know, we are talking about fatty acid synthesis, also known as de novo lipogenesis or DNL. You know very well by now that fatty acid synthesis is a well-fed state pathway which requires a lot of insulin and ATP and acetyl-CoA as well as NADPH. We know that for fatty acid synthesis to begin, you have to make acetyl-CoA available in the cytosol to do fatty acid synthesis. How do you achieve that? You achieve that by pushing citrate in the cytosol. You know how citrate is pushed in the cytosol in a well-fed state, which is not only high insulin, but a high insulin and a high ATP state. You know that in a high insulin and a high ATP state, isocitrate dehydrogenase is inhibited. What happens because of that? Intermediates above the block accumulate because of this inhibition, and citrate is pushed out from the mitochondrial matrix into the cytosol to start fatty acid synthesis. But remember, this is not enough to continue fatty acid synthesis. You still need a downward flow or a forward flow of glycolysis as well as TCA cycle so that you can form more and more citrate and push it in the cytosol to form fatty acid synthesis or to form fatty acid in your body. How do you achieve that? Because in a high ATP state, you know that glycolysis and TCA are slowed down because they are energy producing pathway. So they are slowed down by high ATP inside your cells. Only in the liver there is an insulin PFK2 regulation which bypasses the ATP inhibition on these pathways so that you can turn on glycolysis and TCA a little more times only in the liver by activating PFK1 and increasing glycolysis and TCA and making the availability of citrate in the cytosol. So now citrate is in the cytosol, so let's complete the pathway. Once citrate is in the cytosol, I told you that citrate splits back into oxaloacetate and acetyl-CoA. This is what you need, acetyl-CoA in the cytosol to start fatty acid synthesis. As I also told you that oxaloacetate joins back pyruvate. How? First by giving malate and then it joins back pyruvate. Just to mention, which is not very important, but you must know what's the enzyme working at this step. It is known as malic enzyme. And what does this enzyme do? This enzyme gives you NADPH. Now you know very well that NADPH is essential for fatty acid synthesis because NADPH is a source of hydrogen for fatty acid synthesis. And I told you that the major source of NADPH is your HMP shunt in your body, but the minor source of NADH is malic enzyme too. So it's good to know this step. Once acetyl-CoA is formed, don't forget that acetyl-CoA is only a 2-carbon compound, whereas in your fatty acid you have to form a 16-carbon fatty acid. So you need more intermediates to form a 16-carbon fatty acid from this 2-carbon compound, which is acetyl-CoA. So what happens next is acetyl-CoA is converted into malonyl-CoA. This step is catalyzed by a carboxylase. Carboxylase add carbon as carbon dioxide, and since the substrate is acetyl-CoA, can I label this enzyme as acetyl-CoA carboxylase? Why not? And since it's a carboxylase, it will add carbon as carbon dioxide. So the product is malonyl-CoA, and now malonyl-CoA is a 3-carbon compound, whereas acetyl-CoA is a 2-carbon compound. You must also know that acetyl-CoA carboxylase is an ABC group of enzyme. Very important to know. Why ABC? Because it requires ATP, it requires biotin, and it requires carbon dioxide. So that is why it is also known as the ABC group of enzyme. Are there other ABC group of enzymes that you need to know in your body? Yes, I will enlist them shortly when I talk about regulation, but yes, you should know that ACC, acetyl-CoA carboxylase, number one, is an ABC group of enzyme. You should also know that ACC is the rate limiting step of this pathway. Remember, I told you rate limiting steps are bottleneck of the pathway. Once you block the pathway, the whole pathway can be slowed down. Once you activate this step, ACC, which is rate limiting enzyme, the whole pathway can be activated. So yes, can I now start enumerating the list of lipogenic enzyme? Yes, but remember, first enzyme is ATP citrate lyase, which is the one splitting citrate. So it is called ATP citrate lyase. Where if you have done enzyme classification, you should know lyase. Lyase means that enzyme that splits compound without adding water. So this is the first lipogenic enzyme, ATP citrate lyase. Then comes ACC, which is the second lipogenic enzyme, but it's the rate limiting enzyme. Remember, very, very important. You must know that. But now please understand that with the 2-carbon acetyl-CoA and a 3-carbon malonyl-CoA, your final product, which is 16-carbon fatty acid, has to be formed. So this makes only 5 carbons. So naturally you need more intermediates to come in to do fatty acid synthesis and form 16-carbon fatty acid, which is the first fatty acid to be formed in your body. So how do you achieve that? Though this is a multi-step process by which you form palmitic acid. Palmitic acid is the first fatty acid to be formed. It's a 16-carbon fatty acid with no double bond. A saturated fatty acid is always formed first in your body. And how do you achieve that? With the help of the enzyme known as fatty acid synthase complex. So is it the third enzyme for this pathway, or third lipogenic enzyme? Yes, that is known as fatty acid synthase complex. When I say complex, please know that this enzyme is not a single enzyme, it's a complex of enzyme. Actually, it is made up of 7 enzymes. You don't need to know what are the enzymes of this complex, but yes, it's a multi-enzyme complex rather than a single enzyme. Like ACC is a single enzyme, ATP citrate lyase is a single enzyme. Fatty acid synthase complex is not a single enzyme, it's an enzyme complex made up of 7 enzymes. What does it do? It takes acetyl-CoA, which is 2 carbon, and malonyl-CoA, which is 3 carbon, and forms palmitic acid, which is 16-carbon fatty acid. Now you must know that acetyl-CoA is 2 carbon, malonyl-CoA is 3 carbon. That means there must be the number of acetyl-CoA and malonyl-CoA that enters fatty acid synthase complex to form of 16-carbon fatty acid. Yes, 1 acetyl-CoA and 7 malonyl-CoA. 1 acetyl-CoA and 7 malonyl-CoA join fatty acid synthase complex to give you palmitic acid. Now know that palmitic acid is a 16-carbon fatty acid, but you have long chain, very long chain fatty acid in your body too. So how do you elongate the chain and form a higher order carbon length fatty acid, which is long chain fatty acid? Naturally, if you want to elongate a fatty acid, what should be the enzyme? So this is the next lipogenic enzyme. The next lipogenic enzyme is elongase. Now you must also know that you also have, these are all saturated fatty acid, but you have unsaturated fatty acid in your body. So how do you add double bond and form poly unsaturated fatty acid in your body? By the enzyme known as desaturase. Please also know that you cannot add double bond beyond position 9. So you don't have desaturase beyond position 9. And that is why, so what's the enzyme that adds double bond? Desaturase. This is the last lipogenic enzyme. So to add, because you can have desaturase or you cannot add double bond beyond position 9, that's why those fatty acids become essential in your body and they have to be supplied in the diet. They are known as essential fatty acids. So this completes your steps of fatty acid synthesis. Now, once you've known the step, now let's enlist the lipogenic enzyme, and I will tell you which enzyme is rate limiting of this pathway and which enzyme is regulated in this pathway, and also talk about the key hormones that will regulate them. And as you know, it's a well-fed state pathway. That's why who will activate this pathway? Which hormone will favor its synthesis? Naturally, high insulin. Please know that when I say high insulin, it is always a ratio. That means high insulin over any antagonist hormone ratio. For now, I would say insulin works antagonist to glucagon. So well-fed state means high insulin to glucagon ratio. That is always it is expressed. So this is all about fatty acid synthesis. So just take a look at the whole pathway and understand the overview of fatty acid synthesis. Now what I will do is enlist the lipogenic enzyme and talk about regulation. I'll give you a few seconds to just know the pathway before I talk about regulation. So now, since you know the pathways, now let's enlist the lipogenic enzyme and talk about regulation. So what are the lipogenic enzyme? You already know the lipogenic enzyme, because what is the last segment to be added in this pathway is regulation. Please know that regulation is very, very important and is tested. You must know regulation, otherwise how will you know that when this pathway is switched on in your body and how it is switched on in your body. So let's see what are we talking about now, the last step, which is regulation. So first of all, let's enlist the lipogenic enzyme which do de novo lipogenesis. They are known as lipogenic enzyme. So let's see what are the enzymes that do fatty acid synthesis. They are known as the enzyme for de novo lipogenesis or lipogenic enzyme. First of all, it is ATP citrate lyase. As you know, we started with that, right? What's next is acetyl-CoA carboxylase, also known as ACC. Please, just a note, as I told you, acetyl-CoA carboxylase is the ABC group of enzyme. I'm just enlisting quickly the other ABC group of enzyme, which are pyruvate carboxylase for gluconeogenesis and propenyl-CoA carboxylase for amino acid metabolism. So you should know all the ABC group of enzyme. The first one which I am introducing with lipogenesis is acetyl-CoA carboxylase. Then comes the fatty acid synthase complex. As I told you, it's not a single enzyme, it's a complex of enzymes, 7 enzymes. And then you need to elongate. So came the elongase. And the last one is desaturase. You know that you cannot add double bond, which is desaturate, beyond position 9. That's why there are some fatty acids which are essential in your body. Now, these are all the list of the lipogenic enzyme, but you must know that acetyl-CoA carboxylase is the rate limiting enzyme. As I told you, rate limiting enzyme or committed step or committed enzyme of the pathway. Rate limiting enzymes are regulated, but regulated enzyme may not be rate limiting. Whereas fatty acid synthase complex is the regulated enzyme. So you must know the difference. This is the regulated enzyme of the pathway. First, I'm going to talk about ACC, which is the rate limiting enzyme. You know what ACC does. So let's talk about acetyl-CoA carboxylase and its regulation. So you know what ACC does. ACC, also known as acetyl-CoA carboxylase, it's a ABC group of enzyme. What it does is convert 2 carbon compound into C3, 3 carbon compound. You know what is this 2 carbon compound? It's acetyl-CoA. And you know what's the 3 carbon product of this enzyme? Malonyl-CoA. So first of all, you should know what is the reaction, substrate and product. Now let's see what are the regulation of this pathway. As you know, this is the rate limiting enzyme. Please know there are 2 types of regulation, short term and long term regulation. In short term regulation, one of some few examples I'm using, which is allosteric regulation. That means there are direct positive allosteric activator or inhibitor of this enzyme. Who undergoes regulation? Naturally, rate limiting enzymes or regulated enzymes. Acetyl CoA carboxylase is the rate limiting enzyme, so this will undergo regulation. Which ones? Short term, long term. I will give you example of short term regulation. ACC is directly activated by citrate, because remember, citrate is a big turn on for fatty acid synthesis, because this has to be made available in the cytosol to begin fatty acid synthesis. Acetyl CoA carboxylase is directly activated by ATP. Remember, no synthesis can happen without ATP. What are the inhibitors of this enzyme? Naturally, inhibitor of this enzyme will be AMP, because high AMP inside your cell tells you that you are in a low energy status. High AMP inside your cells tell you you're in a low energy status, whereas high ATP inside your cell tells you you're in a high energy status. Please know that you cannot do synthesis without ATP. Now this is short-term regulation, where allosteric effector like citrate, ATP, AMP, they come in and directly regulate the enzyme. The other one is known as long-term regulation. Long-term regulation is by induction and repression. Apart from this short-term regulation, which is allosteric regulation, which I just mentioned here, there is one more short-term regulation which is known as reversible covalent modification, where the ACC, known as the acetyl CoA carboxylase, is either phosphorylated or dephosphorylated. This is known as reversible covalent modification. And who always does the phosphorylation? Glucagon or epinephrine. Who does the phosphorylation? Glucagon and epinephrine. And who always does the dephosphorylation? Insulin. Please know that insulin always and always dephosphorylates the key enzyme, whereas in glucagon and epinephrine phosphorylate the key enzyme. This is phosphorylated, this is dephosphorylated, this is dephosphorylated form. Dephosphorylated means there is no phosphate attached to it. Now, how does insulin dephosphorylate the key enzyme? By activating which enzyme? So what enzyme is required for dephosphorylation? It is known as protein phosphatase. In a few minutes, I will also enumerate what are the mechanism of action of insulin. So yes, insulin activates the protein phosphatase and does dephosphorylation, whereas glucagon and epinephrine, which enzyme they use to do phosphorylation? They use protein kinase. Protein kinase is used by glucagon and epinephrine to phosphorylate, and insulin uses protein phosphatase, to dephosphorylate the key enzyme. Now, please, as I told you that insulin supports this pathway, that is why which form insulin support? First of all, insulin always and always dephosphorylates the key enzyme by acting through protein phosphatase. But since I told you that insulin or well-fed state turns on this pathway, acetyl CoA carboxylase will be active in phosphorylated form or dephosphorylated form. You decide. It will be active in dephosphorylated form because insulin supports this pathway. So this is all short-term regulation by allosteric effectors or by reversible covalent modification. This is known as reversible covalent modification. I'm sure you must have learned that before starting the pathway. So this is known as regulation. These are all short-term regulation. The last regulation is known as long-term regulation, which is known as induction and repression. Now, naturally, insulin favors this pathway. So what will insulin do? Insulin induces. Insulin will always induce. So insulin induces not only acetyl CoA carboxylase but also fatty acid synthase complex. It induces acetyl CoA carboxylase as well as fatty acid synthase complex and turn on this pathway. So this is all the role of insulin in activating this pathway. Finally, just a quick recap of how insulin can act. Please know very well, though I am just enumerating it, insulin acts by decreasing cyclic AMP inside your cell. Insulin acts by activating protein phosphatase and dephosphorylating the key enzyme, as I just told you, in reversible covalent modification. And insulin is also involved in induction and repression. Induction and repression always take long and days, versus covalent modification and allosteric regulation takes minutes and hours. You should know that about regulation.